So our second type of cardiac myocytes are going to be contractile cells. So if you remember, we had two types of cardiac muscle cells. They went two ways. We had pacemaker cells that were located in the conduction pathway. So they were in that specific area. They account for about 1% of the total heart cells, and they were able to self-depolarize. Okay, now we're on the other side of things, and these are going to be our contractile cells. So these are basically the other 99% of the heart, and these are the ones that are responsible for heart pumping or contraction. Okay, the action potential of cardiac contractile cells is different than pacemaker cells. So the tracing looks different. It still has 3 steps, but it has a plateau portion that allows for time for the heart muscle to contract. So let's take a look at the contractile cells. So cardiac contractile cells have an initial depolarization, which we see here. So here's our depolarization of the contractile cells. And this depolarization is due to fast voltage-gated sodium channels opening. So sodium is going to enter the cell and create that depolarization. Now, another thing to notice too is before we even get to that depolarization, if you look at our resting membrane potential, it's stable. So it's going to have a stable resting membrane potential. This is not like the pacemaker cells. If you remember, they had that unstable resting membrane potential that was constantly depolarizing towards threshold. But contractile cells are different. So we're going to have our initial depolarization due to fast-gated sodium channels opening. Then those sodium channels are going to close, and we're going to reach this plateau portion in our tracing. And so that's going to be this portion here. We can see that the depolarization is kind of extended, okay? And we call this a plateau. So what happens here is that the depolarization that was initially caused by sodium ends up being extended because we have an influx of calcium. Okay, so we continue to have calcium flowing into the cell, prolonging our depolarization. Okay, so it results in this plateau portion of the curve. And then the third phase is going to be our repolarization. So we have this part of the curve here. And this is our repolarization. And basically what's happening is that after a period of time, our calcium channels are going to close and voltage-gated potassium channels are going to open. And we're going to see an efflux of potassium reestablishing our negative resting membrane potential. Okay, and this is from sodium. So we have sodium, calcium, potassium. At this point in time, the calcium, any excess calcium, is going to be pumped back into the sarcoplasmic reticulum and extracellular space. It's basically going to be pumped out of the cell. And the contraction stops. Okay, so here's all 3 phases together. So I do want to highlight, let me just highlight the action potential tracing. It's going to be this one in blue. So here's our depolarization heading up, here's our plateau, and then here's our repolarization coming back down again. Okay, what's also interesting to note on this picture is this line here, this is our tension, and they've just kind of overlaid it on top of the action potential. So this is the tension that's being generated in the contractile cells or the muscle tissue of the heart. So we have the initiation of the action potential, followed by increasing tension and then decreasing tension. And then this is important also. Here's our absolute refractory period. Look how long this period is. It basically lasts the entire action potential. Okay, and so that's our very long refractory period, so that we have no tetany in cardiac muscle cells. It basically lasts the whole length of time, and then it could be stimulated again. Let's say we do another action potential here, and I'm just going to extend this out, and then we're going to generate another contraction here, and then back down again. So we basically have one contraction. We have to wait until the next one can start. Okay, so this is what that's talking about too. The action potential and the contractile phase last much longer in cardiac muscle cells. It's very long. Basically, like, this period of time is much longer than what we see in skeletal muscle cells. And this allows for sustained contractions because we want to make sure that blood is ejected from the heart. This, all at one time. This is very efficient. Okay, and we want to have that efficiency for our heart. We also have this long refractory period, this long absolute refractory period, meaning the heart won't contract no matter what you do to it again, so that we don't have temporal summation. And we want to have a relaxation time between the beats, because that's when the heart fills up with blood. The heart fills with blood when it's relaxed, and that allows for more efficient contractions. So here's the difference. Here's kind of a side-by-side. Here's skeletal muscle and here's cardiac muscle. And you can see here on skeletal muscle, look how short in yellow this absolute refractory period time is. It's very short. Look how long this refractory period is here as well. And this action potential, look, action potential, bam, back down again, repolarization, very short period of time. Look at this action potential here. It's very long and drawn out because of that plateau phase. Very long action potential. Okay, and this is going to have a short action potential. Okay, so just some differences, some side by side. You should be able to compare. We've gone over this many, many times throughout this PowerPoint, comparing skeletal versus cardiac action potentials and tracings. So like we said before, we do have the ability to self-polarize or self-depolarize in the heart, but it does have oversight. So the autonomic nervous system does have an influence over heart rate when needed through cardiac centers located in the medulla oblongata. We have a cardioacceleratory center. So acceleratory means speed up, right? So what we're going to see here is that postganglionic neurons are going to travel down from the medulla, innervate the SA and AV nodes, because those are pacemaker locations of pacemaker cells, and sympathetic nervous system, sympathetic division of the autonomic nervous system is our fight or flight division, and it's going to result in an increased heart rate. You're running away from the bear in the parking lot. So we need to increase our heart rate in order to do that. So that's going to happen through our cardioacceleratory center. On the flip side of things, we also have a cardioinhibitory center. Inhibitory means slow down. And so that's what we see here. Cardioinhibitory center is going to send impulses through the parasympathetic branch of the autonomic nervous system. These fibers are going to travel through the vagus nerve. So if you recall, vagus nerve was cranial nerve 10, and branches of the vagus nerve are going to travel down and innervate the SA and AV nodes. And since this is parasympathetic division, this is now rest and digest. We're going to have resulting decrease in heart rate because when we're laying on the sofa watching TV, our heart rate can be nice and slow, and we're fine with that. But that's through the cardioinhibitory center. And so this is showing you here, this is the medulla, and we've got our fibers traveling down. So here is showing you vagus or cranial nerve 10. This is going to be the parasympathetic input traveling down to the heart, and that's going to be cardioinhibitory. This one here is showing you sympathetic input in green, and sympathetic input is going to be cardioacceleratory to increase heart rate based on fight or flight response. Okay, so one of the things that we can do is we can take a composite tracing of all the action potentials of the heart, and we can generate what's called an ECG, or electrocardiogram. You might even see this referred to as an EKG. They're both the same thing. And these are basically a composite tracing of all of the electrical activity of the heart. So we have what's called 3 deflections in the tracing. When we run an ECG, we're going to have 3 very noticeable deflections. And this is basically where the line kind of changes. So what we see here is our first deflection is going to be this one, this upward deflection. That's going to be the P wave. This area right here is our second deflection, and that's the QRS complex. And then our third one is this one right here, and that's going to be the T wave. Okay. Typically what we see in the clinical setting is a 12-lead ECG. So we place electrodes on the body in all these different locations, and each one generates its own tracing. And then when we put it all together, it looks like our typical ECG that we kind of recognize looking like that. So this is a combination of the electrical activity of the entire heart. So what we can look at with this is a wave of depolarization traveling through the heart. And if you look here in yellow, we know that the depolarization originates in the SA node. And that's what we're seeing here. So we've got initial depolarization in the sinoatrial node. It's basically going to spread from the sinoatrial node throughout the atria. Okay, so we have atrial depolarization, and that's what is indicated on the tracing by the P wave. Okay, and that's what we see here. So that's going to generate this deflection in the ECG called the P wave. Now, we also know that that impulse is going to travel directly through the internodal pathway and is going to activate the AV node. And we know at that point it's also going to slow down a little bit. So that ends up giving us this little flat line here between P and QRS. That's that delay because of the AV node and the bundle of His. Now, the impulse is going to travel through the left and right bundle branches, right? And then it's going to travel up through the walls of the ventricles through the Purkinje fibers. And in this way, we now have ventricle or ventricular, let's see, hold on. Okay, ventricular depolarization. That's going to spread throughout the ventricles, and that's going to give us, let's back this up. Here we go. Ventricular depolarization, which is going to give us our QRS complex. And it's much more complicated than the P wave, just because those ventricles are much larger, and it gives us a more complicated composite action potential tracing. And so that's basically the wave of depolarization. And so we can see here with our particular QRS, here's our P wave. This is responsible for atrial depolarization. So we have a wave of depolarization that's going to travel from the SA node, because that's where it originates through the atria. Following depolarization, we're going to have contraction. So P wave is going to consist of atrial depolarization, which is going to be the electrical event. And then that's going to be followed by atrial contraction, otherwise known as systole. That's the fancy word for contraction. And that's going to be the mechanical event. So that's actually the heart contracting. This is important. The electrical event always happens before the mechanical event. Those cells have to have the signal first before they can actually contract. So here's our electrical signal. Here's our mechanical contraction that's going to happen as a result. Now, during the QRS complex, we're going to have ventricular depolarization. Okay. Shortly after that, we're going to have ventricular systole or contraction during that time. And that's what's shown by the QRS complex. Now, at the same time, we are going to have atrial repolarization and atrial diastole, which means relax, okay? And that's happening at the same time, but this ventricular depolarization, ventricles are so big, this waveform kind of overshadows it, so you don't really see the specific events that are happening in the atria. And then the last deflection is our T wave. And so if you recall, during QRS, we had ventricular depolarization. Well, now it's time for the ventricles to reset, to get ready for the next time around. So we're going to have ventricular repolarization during the T wave, and this is going to be accompanied by ventricular diastole, or a relaxation of the ventricles. So it's very cyclic. This is going to happen over and over again. But if you think about it, the atria are going to contract first, followed by the ventricles contracting at the same time. And all of that is preceded by the electrical events. So the wave of depolarization is going to spread through the atria first, and then it's going to spread through the ventricles. And so we've got a couple other segments here, like the PR interval is going to be this period of time right here. So this is basically from the beginning of atrial depolarization to the beginning of ventricular depolarization. And we do have this period of time here. This is kind of that delay, that flat line between the two, the delay at the AV node. ST segment is kind of drawn out because we have the plateau phase of the action potential of the contractile cells. And then QT interval is basically the whole time of ventricular depolarization followed by ventricular repolarization. This kind of is an introduction to cardiac cycle, which is going to be our next topic. But just as a reminder, the mechanical events in the heart, so either mechanical events means either contraction or relaxation. And they are always going to follow the electrical events. So the electrical event has to happen first. That's the stimulus for the mechanical event to follow. Some vocabulary. Systole means contraction, and diastole means relaxation. And basically, one cardiac cycle means all of the events that occur during one heartbeat. And this is going to be atrial systole followed by atrial diastole, and then ventricular systole followed by ventricular diastole. So the atria contract together and the ventricles contract. This is just a little chart to remind you what's happening during each one of the deflections in the ECG. So I've paired it up. Here's the electrical events that are happening, and here are the mechanical events. And we've got to travel through all of this creates one cardiac cycle. And the rest of this we're going to get with our cardiac cycle lecture.